Okay, now let's start discussing these part 2 questions. So the first question, plants are important for the survival of organisms. Therefore, man propagates useful plants using various methods, right? So we have learned the plant propagating methods. You have natural propagating method and the artificial. So here they have given um, about the natural vegetative propagation method, right? So they have given two examples. We have to find uh, which type of vegetative method is that. Okay. So normally the asexual reproduction is divided into two as natural vegetative and the artificial vegetative propagation. Okay. So in the natural vegetative propagation, we have roots, the propagation done by roots, leaves, suckers, runners, bulbils, and also the underground stems. In artificial, we have rooting of stem cutting, grafting, layering, tissue culture, right? So those are the main things. So here they have asked the natural vegetative, right? So they have given Akapana. Akapana, they propagate through their leaves. Their if you are telling in Tamil, that is Sadai Karacha, in Sinhala Akapana. So that is done by leaves. Then pineapple, that is done by the bulbils. So you know bulbils, that is the special reproductive structure formed by a modification of a vegetative bud or a flower bud. So a bud or flower bud will give the bulbils. So that is given like pineapple, jute, hondella. All those are examples for bulbils. When you consider for leaves, you have begonia. Other than this acapana, you have begonia, queen of the night, or you call kadupul. Okay. So when you're writing the discussion, you can all write all those things, right? Then they have asked write two advantages of tissue culture. So I told tissue culture, that is a method of artificial vegetative propagation, right? So you can write any of these as the answer. So when you are doing tissue culture, there you can produce offspring which are identical to mother, right? All the babies are identical to the mother. Then you can propagate large number of plants at the same time. At one time, we can have large number of plants. Then what else? Next one, you can say they have plants in short time. With less time, we can have large number of plants, right? And also within a limited space, we can have a healthy plant. Okay, then by using hybrid tissue with favorable, you can exchange the G and we can make a favorable plant. Okay, plant or species. So any of these, in this, any two examples will be your advantage will be the correct answer. Okay, so this is a part of reproduction. So that is connected next with the genetics, heredity, right? Okay. Now see, Gregor Mendel explained the transmission of inherited characters during sexual reproduction through his experiments. A pure breeding tall bean plant, it was crossed with pure breeding short plant. So they have give dominant feature T, tall as capital T and the short as simple T. Right? So the first question they ask, what is the genotype of the plants? So we have two things. One is genotype, other one is phenotype. So when we talk about first phenotype, phenotype is the external feature that you can observe, right? You can externally observe like here tall, short, height, then the color. Those are the external feature you can see, right? Color. Then the if it is a human, then you can say eye color. Likewise, external feature we call phenotype. Okay. Genotype is the gene composition to determine that feature. Which gene determines the height? Which genes determines the color? That is what we call genotype. Okay. So write the genotype. So here the genotype is tall. That is one dominant character, tall, tall. Other one is TT, short. So if you have two recessive character, they will give the short. If you have two dominant character, they will give the tall. So the genetic composition, that is you call genotype, you can state like capital T, simple T, simple T. Right? Then state the gamete formed by the each. So these are the parents. So from that, the gamete from capital T, we will have T, and simple T, we will have T. So that is also simple. That is the gamete. Already, I think we discussed in the MCQ. Right? Now they are asking, find the following details of F2 generation using Punnett square. Normally for Punnett square is done by the, like F2 generation, you can do with the Punnett square. So what you have to do, you have to select one side male, other side female. Okay. So we are crossing with male and female. So here you will have a gamete T, capital T, simple T this side, capital T, simple T the other side. So you cross TT, then first you will get tall one. Then you capital T, simple T. Again, T is the dominant character, you get a tall one. Then simple T, here simple T, simple T. So that is here short. And the simple T, capital T. This is tall. Okay. So now this is the Punnett square. This is an easy way of making a Punnett square. See, T, T, then you everything you have to join. T, T, this T. Then again this T with this T with this. Okay. So here capital T will give the dominant character. Here also you have a capital T, so dominant character, simple. So that is the recessive character. Now they are asking the genotypic ratio. So first I'll talk with phenotypic, that is the external feature. So external feature you can see 3 tall plants and 1 short. So you put the genotypic ratio 3:1. Okay. And the genotypic ratio, that is the genetic. See, this is capital T, capital T. You have only one capital T, capital T. Then capital T, simple T, capital T, simple T. You have 2. So 2. Then simple T, you have 1. So 1:2:1. So that is the genotypic ratio. Okay, is that clear? Now the C part. Following is a longitudinal section of a heart. So we are going to name. What is the first one? Here you can observe this one. So that is pulmonary artery. Right? Then second here, this part, you can write superior vena cava, right? Then third one here, pulmonary veins. Then fourth, this is right atrium, and the fifth, that is left atrium. So here in a heart, the upper part of the heart we call atrium, the lower two part we call ventricles. And when you divide like this side right and this side left. Okay, with that you have to name. So you have 4 chamber in human heart, right? So these upper two are auricle, lower part ventricle. This side right side, left side. Okay, right. Next is write the difference between 1 and 2. So normally in a heart, arteries take the oxygenated blood. Normally the function of arteries, they take the oxygenated blood. And function of veins, they takes the deoxygenated blood. Right? So these are the functions of arteries and veins. So here the first one is pulmonary artery, and the second, not the second, So the question should be write the difference between 1 and 2. So 2 is the superior vena cava. It should not be 2. Here you should write the difference between 1 and 3. Okay, the question should be 1 and 3, not the 2. Okay, 3. Okay, so 3 is pulmonary vein and the first one is pulmonary artery. Okay, so normally arteries take oxygenated blood and veins take deoxygenated blood, right? But in the heart, what happens? The superior vena cava drains the blood to right atrium. From the right atrium, blood is travel to right ventricle. This blood is deoxygenated blood. So that is why I'm showing in blue. And this deoxygenated blood only carried through this pulmonary artery to lungs. Okay. Then they reach the lung. Here, let's take, they reach the lung. They make the deoxygenated blood as oxygenated, right? From there, they take the oxygenated blood through the pulmonary veins to the left atrium. Okay, so that is the concept. Normally, arteries take oxygenated, veins take deoxygenated. But in heart, the pulmonary artery, they takes the deoxygenated blood and pulmonary veins, they takes the oxygenated blood. Okay, so pulmonary veins take oxygenated and artery deoxygenated. That is verse wise. Okay, so that is the answer for the second question. Then the next one, what is the function of valves? So these are the valves. Here you have this valve, we call that as the semilunar valve. That is present here, these two. The opening of the aorta and the opening of the pulmonary artery, we have the semilunar valves. And here in the left and the right, this side, right and the left part, between the auricle and ventricles, we have the tricuspid and the bicuspid valves, right? So the main function is to prevent the backflow of the blood. When blood enters from right to atrium to ventricle, they should not move back to the atrium. To avoid that, they have the cuspids, valves, right? So the main function is to prevent the backflow of blood, right? Then state the system and the organ where the blood is transported by the vessel 1, right? So that is we call pulmonary system. So there are heart can carry out two system you call double circulation. So one is to the lungs, the other one is to the other parts of the body. So to the lungs we call pulmonary circulation, and for the other body we call systemic circulation. So here the one that is the pulmonary artery, they take the blood to the lungs. So that is called pulmonary circulation. So the system pulmonary circulation and the organ lung. Right? Then the next question, explain how the lub sound in heart is produced. Okay, so the lub, heartbeat produce lub sound. So whenever the tricuspid, these two tricuspid and bicuspid valves are closing, it gives the lub sound. And whenever the semilunar valves closing, then it produces the dub sound. Okay, now let's move on to the chemistry part. So here, this is a iron extraction method. here they have given a blast. You call this as the blast furnace that is used to the extraction, iron extraction. So normally the raw materials that are used is limestone, coke and hematite. So hematite, the formula Fe2O3. So they are added to the blast furnace through this way, right? Then what is added from 2? So from 2 we add hot air blast. So all these air goes in, and with these raw materials they start to react, and at number 5 only we get the iron, molten iron things, and at number 3 we get the slag, that is the impurities, right? So what is added to from 2? That is hot air blast. What are the materials given 3 and 5? So 3, that is you call slag, and 5, that is the iron. We are extracting iron, so iron is extracted there. Then write a reaction taking place in the furnace produce carbon dioxide. So here you are adding coke. That coke reacts with the oxygen and they form carbon dioxide. Okay. And also during the iron, when hematite reacting with the carbon monoxide also, carbon dioxide is formed. Right? Then the next is write the reaction in the furnace which produce 5. So 5 is the molten Fe. So this Fe is produced by this. The hematite that is reacted with carbon monoxide and they give the iron with the release of carbon dioxide. Okay, so that is the reaction. So the reactions are very important. Okay. Then the B part, there you have PQR given below. Crystalline solid, okay, that is the first hint they have given. So we don't know what solid is that. And dissolve well in Q, right? So you should find what is Q first. So let's go to Q. That is a colorless liquid, right? Their boiling point is 100. So you know water only boils at 100. So this Q is water. So they have told this P can dissolve well with Q, means they can dissolve well with water. Therefore, if something can be dissolved well with water, we call that as the polar substance, right? Okay. Now R, that is a volatile liquid. Yes, they can evaporate and they have very low boiling point. Okay, they can evaporate but with very low boiling point, and they does not dissolve in this P. Okay, so when you consider P, they are not dissolving with that. So we can say that is a non-polar, right? So based with these datas, now let's move on to the question. What is solubility? So solubility, you have to know the definition. Solubility of a solute at a given temperature can be defined as a mass of that solute. You are considering mass of that solute dissolving in 100 gram of solvent at a certain temperature. So you should know the temperature. At certain temperature with 100 gram of solvent, the mass of the solute is called as the solubility. Okay, so the factors affecting solubility, we have 3 factors. One is temperature. When you increase the temperature, solubility differs. Solute, when it changes according to the difference. If the solutes are different, solubility different. Then if the solvent are different, the solubility also different, right? So solvent is the large amount in the solution. Solute is the smaller, lesser amount in the solution. So solute plus solvent will give the solution, right? Now they have given crystals of P contained other impurities in small amount, right? So here in crystals, they have impurities. Now that is were dissolved with Q and they have increased like 80-degree temperature with highly concentrated solution. So we call that as saturated solution. You are making the solution saturated. By providing heat, you are making the solution concentrate. Then what you are doing, you are filtering that solution and keeping for cool down. So what we are doing, already it is in crystal and they have impurities. So in a crystal, if you want to remove the impurities, you have to saturate more and cool down. So then what you can get, so if you cool down, you can get a pure crystal because here you have impurities. Impurities means that is with the unnecessary particles. By cooling down, by filtering, by heating, you are making the crystals pure. Okay, so the answer, the observation when the filtrate cool down, you get pure crystals. Okay, right. Then what do you call the process mentioned A? So this making this already this crystal with the impurities, if you are making it pure, that is called recrystallization process. Okay, that you have learned in the grade 11 unit 3. Right. Now we have the physics part. Right. Here, a rubber sucker held on the wall is shown in the diagram. Right. How is the rubber sucker hold onto the wall? So how it is hold here, this part, there is a wall, this is the sucker, by the atmospheric pressure. Due to the atmospheric pressure, they are hold. How can be the air pressure P and Q position is held pressed? So how can be the pressure? So the pressure of inside, it means Q, pressure at Q, here this is, I have shown here, right? There the pressure will be less and here the pressure will be more. That is the outside pressure will be more positions. That is why the air is pressing this inside, so that is not falling down. Okay, so outside air atmospheric pressure is greater than the inside air. Okay, here you have less pressure than the outside. Then what is the instrument used to measure atmospheric pressure? So that is what we use normally, barometer, to find the atmospheric pressure. Right. Then the next part, life has become easier with the use of different types of electromagnetic waves. Write down main characteristics electron. So you know waves can be divided into two. One is mechanical, electromagnetic. Mechanical waves can be divided into two. One is transverse, the other one is longitudinal wave, right? So longitudinal wave, the particles and the wave, they propagate parallel to each other. Transverse wave, the particle travel perpendicular to each other. Okay. Then the electromagnetic, before that transverse wave, they need a medium for a propagation. But electromagnetic waves, they do not need a medium. So mainly, if a wave traveling with a medium, that is transverse. Without a medium, electromagnetic. So main character, you can see no need of a material medium. Then what else? So when you take EM, electro and magnetic. So they will have electric and the magnetic like this with the perpendicular direction. So if you give any electric and magnetic external, if you give any electric and magnetic source, they are not affected. Okay, so that is another characteristic. And you can say here, the light, they can travel in 3 into 10 to the power 8 meters per second. That is also another feature, right? Okay. Name the electromagnetic waves used at the following instance, right? To find the fractures of the bones of the body. So normally, if you have any body fracture, bone fracture mainly, we do by the X-ray. So what are the EM wave rays like? Uh, Gamma rays, X-rays, UV, visible, IR, radio, micro. So those are the types of wave. So you have to know all what is the advantages and also what are the disadvantages. Right. So normally if any fractures, we use by X-rays. If you want to find some diseases using heat, for that we are using IR. Okay, IR rays, right? To identify fraud currency, that is UV. So if any in the currency, if any that symbol cannot be observed, you can send the UV and you can find the symbols there, identify the currency. Okay. So those are the main things.